Welcome back to the second part of the webinar. May we now invite Professor Song Xinyuan, the chairperson of Department of Statistics, to introduce our next speaker, Professor Yingying. Professor Song, please.
Okay, thank you. Good morning. I'm delighted to introduce my colleagues, Professor Yingying. Professor Wei is an associate professor in the Department of Statistics. She obtained her bachelor's degree in mathematics from Tsinghua University and her MSc engine degree in computer science and a PhD degree in biostatistics from Johns Hopkins University in 2014. Her research focused on developing statistical methods for analyzing noise, complex, and heterogeneous big genomic data. Her 6 bioconductor R packages have been well received by the community, with more than 69,000 downloads to date. Professor Wei received the Faculty Explanatory Teaching Award from Faculty of Science, CUHK, in 2017, and the W.J. Uden Award in Interlaboratory Testing from the American Statistical Association in 2019. Today, Professor Wei will present a talk entitled Differential Inference for Single-Cell Sequencing Data. Without further ado, let's welcome Professor Wei. Professor Wei, please.
Thanks, Professor Song, for the very kind introduction and the faculty for the precious opportunity. First, I want to apologize that my network connection is not very stable today. So sometimes there may be several seconds delay, but hopefully overall it is fine. Sorry about that. So today I will talk about differential inference for single-cell RNA-seq data. So I will first briefly review the main problems associated with the general analysis of single-cell RNA-seq data. Then I will go to the main topic for today. And finally, I will discuss the future work. So in the past, when we do gene expression data analysis, we use the bulk gene expression studies. The idea is we will collect tissues from the patients, and then we will, which contains thousands of cells. Then we will scramble those cells and measure an average gene expression levels for those cells. With the single-cell RNA sequencing technologies, now we can dissociate the tissues into individual cells and measure the gene expression levels of each individual cell, which provides us with unprecedented opportunities to study many biological systems at the cellular resolution and for us to understand the cellular heterogeneity. So at the end of the day, usually we'll get data like this, where each row corresponds to a gene and each column corresponds to a cell. But unfortunately, the cell type labels for each cell is unknown. So our goal is usually we need to cluster the cells into cell types to discover new cell types. And the problem is usually we need to collect the data in multiple batches, so which creates a lot of problems for the data integration, and which has become a very hot topic and received extensive research in the past several years, which is the meta-clustering, which means the joint clustering of samples across the batches for single-cell RNA-seq data. So here I will briefly review the main challenges. The most important challenges are the batch effects and the dropout events. So people have been aware of the batch effects in industry a long time ago. So for example, you can imagine for those bottles produced in January, so they may contain 505 milliliters, while for those bottles generated in February, they will contain 498 milliliters. So as a result, there will be a 7 milliliters difference between these 2 months. So that is called the batch effects. And for single-cell RNA-seq data, so people have seen that the magnitude of the batch effects can be comparable to the difference between cell types. And you can see that the technical artifacts actually vary from gene to gene, which creates troubles for the analysis. So, and meanwhile, there's another phenomenon called the dropout events, which means a gene is actually expressed, but unfortunately, due to the sequencing technologies, we fail to detect them in our experiment. And people have found that actually more highly expressed genes are less likely to suffer from dropout events. So in statistics, that is called missing node at random. You can imagine if you do a survey to ask about people's income, so then those higher people with high income may more likely to tell them their income, while those lower income people may just tell you their income is 0. So we need to adjust for these bias in our data analysis. So as a result, so you can see there has been very extensive research on how we can do the joint clustering of single cell RNA-seq data across batches in the past several years, and including ourselves. In our publication, we've shown that our proposed method at that point outperforms the state-of-art methods at that point in terms of the clustering accuracy. So today I will move on to the differential inference for single cell RNA-seq data, because I think in the past when we do the cell type clustering, we usually ignore whether the cell was collected from a healthy individual or a diseased patient. But however, I think for most biomedical research, we want to compare, right, across conditions, either case or control, or across developmental stages. So the problem is that although we don't know, right, the cell type of each individual cell, but we know whether the cell was collected from an individual who is a healthy control or a case, right, a disease status. So then the idea is to say, okay, how we can identify those genes that are differentially expressed between the conditions for each individual cell types, right? For example, here the yellow cell type, you can imagine it may correspond to the beta cells for patients. Then you can expect some genes may be differentially expressed between healthy individuals and the type 2 diabetes patients for the beta cells, right? So similarly, we want to do it for the green cell type, right? So the purple cell type. And meanwhile, the data are once again collected in multiple batches. So we have technical artifacts. And moreover, the cellular composition will also change between conditions, treatment conditions. For example, you can imagine when the person is ill, so then the proportion of T cells may increase. So how can we do the inference? So maybe we can view this problem as an analogy. So it has already been a classical example in data science, right? So that we can cluster customers into different groups according to their consumption behaviors, although they don't tell us their group labels. So here, the problem of interest, you can imagine, right? So in the pandemic, maybe the food consumption is really important, okay? So then what we care about in this problem is to say, we can group customers into, say, 3 groups: high income group, middle income group, and low income group. Then we want to know for each separate group, right? So whether there is any change in their consumption of each item of food, right? So whether, for example, the consumption of potatoes has increased or decreased for high income people. And once again, we care not only about one city, say Hong Kong, right? We also care, for example, other cities like Shanghai, right? And we can expect there are location differences between these cities. But nevertheless, right, so we care about, in general, for the high income group, right, so what will be their change in their consumptions due to the pandemic, right, across cities. So that is a problem. So this is what we want to infer in these studies for the cells. And just like the customer groups are unknown, so here the cell types are unknown, although the disease conditions for each cell is known. So traditionally, how people do differential inference for gene expression data, for the bulk data, so there has been very successful methods such as Lima for microarray, so HR and DE-seq for RNA-seq data. So you can see all of these 3 papers have received tens of thousands of citations. So for your reference, so most of the statistical journals, their impact factors are below 5, so including the very top ones. So you can see how impressive these numbers are. So I always admire this work very much. So I think why they made such high impact, because they address problems that biomedical researchers really care about. But however, these methods are not applicable to single cell RNA-seq data. The problem is that for the bulk data, each column corresponds to a patient, and you know whether the individual is a patient or a healthy individual. But for single cell RNA-seq data, now each column corresponds to a cell, and the cell type labels are unknown. So we need new methods. So recently, because the problem is very important, hopefully I have convinced you. So there has been efforts that try to do the differential abundance inference for single cell RNA-seq data. So the idea is to say, okay, whether the cellular composition has changed over conditions. For example, you have seen increased amount of T cells. But however, this method, they need to assume the cell type labels are known, which is unrealistic for single cell RNA-seq data, or they need to do the clustering and assume that the gene expression profiles of cells of the same cell type will stay the same across conditions. In other words, they need to assume, for example, beta cells, the gene expression levels are the same between healthy controls and type 2 diabetes patients, which is unrealistic. And meanwhile, people really need to do differential gene expression detection analysis, but solid statistical methods are lacking. So the current approaches are multi-stage approaches. So first you will do normalization, assume it is correct. Then you will adjust for the batch effects. Then assuming the batch effects has been adjusted, then you will impute missing data due to dropout events. And then you will do the clustering of the cell types by ignoring the treatment condition, whether the person is a healthy or a diseased patient. And then after you do the clustering, right? So then for each cluster, you will distinguish between the treatment conditions and then try to do differential gene expression detection. However, this is self-contradictory because when you do the cell type clustering, you assume there's no differentially expressed genes. That's why you can do the clustering. But when you do the differential gene expression detection, you need to assume actually they are different across conditions. And moreover, when you have these multi-stage approaches, the errors at each step will be propagated to the downstream analysis. So the statistical uncertainties will be ignored. So our goal here is that we try to provide statistical methods that can provide rigorous inference to do both the differential abundance inference and the differential expression inference, and meanwhile address all the other issues. So we propose a hierarchical model built upon our previous model called the Basin. So here we'll briefly introduce the model. So we've assumed for the ice cell of the T's treatment collected from the B's batch, its probability of belonging to the K cell type is equals to a batch-specific proportion of cell types. Okay, so then given the cell comes from the K cell type, we assume its underlying true gene expression levels will follow a negative binomial distribution whose mean can be decomposed into a baseline gene expression level and a cell type effects and the treatment effects, and then a batch effects, as well as a cell-specific normalization constant. But unfortunately, we will not be able to observe these underlying true expression levels all the time. So we will include an indicator to see, okay, whether it's a successful detection or it's a dropout event. And we let the dropout rate to depend on the underlying true gene expression levels. Finally, suppose it is a successful detection, so then we will observe the underlying true gene expression levels. If it is a suppose it's a successful detection, we can observe that. Suppose it is a dropout event, then we will observe a 0 instead. So we only observe the data in the gray box, but fortunately with statistical techniques, so we can learn all these other latent variables and parameters. So in this study, so the parameter of interest are the treatment effects, as well as the cell type specific, batch specific proportions. So, but however, there's another thing we need to pay in mind is that not all experimental designs will work, right? So suppose, for example, if in batch 1 and batch 2, you collected only treatment cells from treatment 1, in batch 3 and batch 4, you collected cells only from treatment 2, and batch 5 or batch 6, you collected cells only from treatment 3. So then there's no way you can separate those technical effects from cell type effects from treatment effects. So our theoretical results show that suppose we can construct a batch graph, so where each batch corresponds to a node, and 2 batches will share an edge if they share cells collected from the same treatment. One treatment is enough. And that this graph is connected, then it's a valid experimental design. So then we can develop algorithms to do the statistical inference for both a parameter point estimator as well as a quantification of the uncertainties. I won't go into the details. So another challenge here is about computation, because now we can easily have tens of thousands of cells. So how can we do fast computation? Here I want to illustrate the idea rather than the details. Please allow me to deviate a little bit. So may I ask a question, right? So suppose we want to know the average salary in Hong Kong. So how can we do it? So the first approach is that we can do a census and ask each person what is his or her salary. Then we add these 7 million numbers up and divide it by 7 million. So you can see that we need 7 million computations. So one way to do that is we can resort to parallel computing. For example, we can establish a computing center in each of the 18 districts in Hong Kong. So then after calculating the average for each computing center, then we can aggregate these numbers. And another approach is a statistical approach, right? So rather than asking 7 million people, we may ask only 10,000 people. So then we can have a pretty good estimate of the average salary in Hong Kong. So that is the beauty of statistics. And actually, so nowadays, the most popular machine learning algorithm, deep learning, its core component is a so-called stochastic gradient descent, which is exactly at each step, you will subsample a subset of all the data, right, to calculate the gradients. So that's a statistical component, as well as a parallel computing with a GPU programming. So actually, in our algorithm, we also borrow ideas from these approaches, although not exactly the same. So as a result, so we can actually determine the number of cell types from the data and detect those genes that are differentially expressed between cell types, and as well as those for each cell type, which genes are differentially expressed between conditions, and also do the differential abundance analysis for cellular compositions. So we conducted simulation studies with 3 treatments and 6 batches. So these are assumed cellular compositions. And so we are actually be able to recover the assumed truth for both the cell type effects, treatment effects, the underlying gene expression levels, and batch effects are pretty well, and discover the number of cell types. So our algorithm outperforms the state-of-the-art method on the clustering accuracy. And as you can see, right, so suppose the 2 cellular compositions, they have no difference. So we won't reject the null hypothesis, but there is an overall difference. So then we will provide a significant p-value. And meanwhile, once we see there is an overall difference in the overall distribution, which we call the global test, so then we can also perform element-wise test to see, okay, actually the cell type 4 is differentially abundant between these 2 treatment conditions. So you may say, okay, for simulation studies, it always works, right? So I think it's encouraging when we analyze the benchmark data sets that has been analyzed many times in the single-cell RNA-seq data analysis literature. So we can see our proposed new method that considers the treatment condition for each cell actually not only improves our own algorithm before substantially, as well as the latest also other algorithms that also updated their algorithms. So I think the result is quite encouraging. So, and also we can see, right? So for these type 2 diabetes patients, we can see actually we can do the differential abundance inference, and we can see actually it is the beta cells so that are differentially abundant between healthy individuals and type 2 diabetes. So we also do the detection of intrinsic genes, which means the genes that are differentially expressed between cell types. So our algorithm identifies about 500 genes, which is a reasonable number. But those multi-stage wise approaches, they pretty much will call all the genes to be differentially expressed between cell types, which is unrealistic. Because our method is also able to do the cell type specific DE genes. So then for each separate cell types, and we also do for those differentially expressed genes, we do pathway analysis, and we can see all these learned enriched pathways are related to metabolism or pancreatic secretion. So the results are quite encouraging. So as a summary, so for this part, I think we propose a hierarchical model so that can simultaneously try for batch effects, cluster cell types, impute the missing data, and there's no need to do normalization or any preprocessing. So that we can do both the rigorous differential expression inference as well as the differential abundance inference to detect those genes that are differentially expressed between cell types, as well as for each cell type, those genes are differentially expressed between conditions. So what would be the next step, right? So here, this will be the future work plan. So the first thing I think was very exciting is about the temporal data. So here, we now assume there's a case control condition. Actually, our algorithm can work with multiple conditions. So one thing is you can view the multiple time points as multiple conditions. But nevertheless, for temporal data, so actually there can be smoothness across the time points. So then we can further build in statistical modeling to gain efficiency. So actually, Professor Ting-Feng Chen and Professor Jerome Hui from School of Life Sciences has currently collected data for developmental stages. So here we illustrate our current approaches on the Drosophila lag discs. So they have collected data for 3 developmental time points. So as a result, you can see, for example, the purple cell types, right? As the Drosophila lag continues to develop, right? So the purple proportion decrease while the blue proportion increase. And then for each cell type, we can see which genes are differentially expressed over time, right? For example, here for cell type 1, you can see some genes, once they are turned on, they are always on, right? So some genes, however, when they are turned on, they can be off later in the 3rd time point. And not all cell types have differentially expressed genes across time. For example, the cell type 5, there's no genes that are differentially expressed across time points, so they will stay the same. And another thing is that currently it's very hot topic for the spatial genomics. Many people are working on how you can integrate spatial genomics with the single cell RNA-seq data. So I think we have a niche here, but not many people were looking to temporal data. I think we have a niche here, right? Because how you match the cells across time points is also challenging. As we say, the cellular composition can change over time points, as well as genes, right, of the same cluster can also differentially express across time points. So I think we have a niche here built upon our current work. So our ultimate goal is hopefully we can build some 4D instead of those currently 3D cell atlases for some developmental systems. And another generalization is currently we focus on single cell RNA-seq data, but for single cell sequencing technologies, there can also assay different modalities. So in the future, we will also generalize our algorithms to multi-omics data. And I think this is I really enjoy the collaborative work. So we first initiated the idea of DeepSeq together with Professor Kevin Ip from computer science. So actually, Kevin also gave a talk in last year's faculty research day. So both of us work with clinicians. So Kevin works with colleagues from Faculty of Medicine on liver cancer single cell RNA-seq data, and I work with colleagues from Fuwai Hospital in Beijing on cardiovascular disease single cell RNA-seq data. So we are aware of the problems. So then we discuss, okay, how we can address that. So later we work together with my former PhD student, Fang Da Song, who's now an assistant professor at CUHK Shenzhen, to develop the method.
Okay, Yingying.
MPhil student Tom, who do the analysis for the Drosophila. Yeah. So I will. Yeah. So maybe I will continue. So hopefully the network will be back. You know, for the collaboration within Faculty of Science, so I've talked about the development of Drosophila. So actually, Jerome has also collected single cell RNA-seq data for jellyfish for 5 time points. So with more time points, so we are actually currently working also on this data set. And so in the future, we expect, so we will collaborate with Professor Zhixiang Lin in our department on single cell multi-omics data. And once we can learn the 4D cell atlas for the organisms, so we are expecting we can work together with Professor Yingying Wu, who's an expert, to understand, for example, how the cells are organized and interact with each other in the tissue. So finally, I want to thank the fundings and especially the CRIMS. So that's all. So thank you very much for attention. And I guess maybe there are any, I think that maybe there have been network problems, right, Professor Song?
Thank you for the excellent talk. So first of all, I'd like to ask questions from audience. Any question?
I see there's a question in the chat, right? So the question is to say, okay, do we have the same cell types in both control groups and disease group? What happened if the cell types are different between 2 groups of data? Okay, so I think that is a superb question. So currently in our current work, as well as those literature I just mentioned, so we assume the number of cell types will stay the same in control group and disease groups. But nevertheless, our method and also the theorem can be generalized to scenarios. So where the disease group can have a new cell type that you haven't seen before in the control group. So for example, like for cancer tissues, you can expect, right? Maybe you will see cancer cells in the disease group. But I didn't show the results here, so it is doable. Yeah, so that's a fantastic question.
Yeah, thanks, Chunshan, for raising the question. Any other question?
Yes, I have one.
Yeah.
Yingying, I think that this is very interesting, and thank you. I remember that shortly after I joined the CUHK, I heard one of your earlier presentation. I become quite interested in using the statistical method down to the single level. And I will say that again, I think this has been very interesting and very significant progress. I was wondering that, with your methodology development with this differential inference, I noticed you applied mostly on the pancreas data, and the data source and reliable data source are very critical for you to do this analysis. I wonder that methodology-wise, given that if you have the single cell data from other parts of the cells, for example, from on the kidney or on the liver, methodology-wise, you'll be able to apply that as well, right? Or you need to develop a new set of analytical tools.
I think, thanks, Dean, for the fantastic question. So currently we are applying to the same tissue under different conditions. So our currently work is, I think this is also related to the question Chunshan mentioned, is to say the cell types, the number of cell types will stay the same across conditions. But we can expect, suppose we want to compare, so the liver to the kidney. So then you will have some kidney-specific cell types and liver-specific cell types. So then I think we will need to study the theoretical conditions a little bit carefully. And I think it's doable, but there will be assumptions. For example, they need to share some cell types like T cells or B cells. Suppose we have two tissues, their cell types are completely different. So then, for example, eye or maybe the skin, I'm not sure. So suppose they don't share any cell types, then I think we will run into trouble. But suppose they share some cell types, so then I think it's doable. But we need to study the theoretical properties very carefully for how many cell types they need to share. So whether two cell types or one cell types will be enough. Maybe one is not enough. Maybe we have two or three. So yeah, but that's a fantastic question. We need to further look into. Yeah.
I see. Thank you. A basic clarification also. You suppose someone provide you with an obviously tons of data. How do you actually get the data incorporated into your analysis? Do you design a program that will automatically pick the data, then that you incorporate the different analytical methodology, or your student have to somehow separate the data into different sets and somehow input into your data analysis?
Yeah, so I think when we get the data, yeah. So for the methodology analysis, usually when we develop the computational algorithm, so usually because we need to do benchmark studies, so usually we use the public available data. So there are large public data repositories like the GEO where you can download the data. But for collaborative, for example, for Jerome and Tingfeng, they will kindly also provide us with their data. But no matter for public available data, for all our collaborators' data, so we need to understand how the data were collected. So as Dean just mentioned, so then we need to see how the data were actually collected, whether they were collected in multiple batches or they were collected together. So that's something we need to, the students and I need to look into details to understand the experimental design of the data sets. So after we understand that, then we can do some pre-processing, then we can apply our algorithms and methods. Do I answer your question?
Yeah, thank you.
Yeah,
okay, thank you, Dean, for raising a question. Then KB, please raise your question.
Okay, Professor White, I think it's a fantastic talk, and you have a lot of collaboration with people in the Faculty of Medicine and biologists in our school. Just one question, I like to share your experience, how to train students, or graduate students or undergraduate, who can actually speak both languages in your lab, right? That 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 can be very good at statistics and also understand the problem. Single cell sequence is very hard, recently. So yeah, I would like to share how you do it.
Yeah, I think maybe I'm more familiar with how to train my students who is a math or stat background with the biological ideas versus the other way around. Maybe Jerome and Tingfeng and other colleagues from School of Life Science has more experience. I think they have fantastic students who can work superbly with bioinformatics. And also, because we have a bioinformatics journal club, you can see many students from School of Life Sciences also have very deep insights in the data analysis. For my students, I think at first, I think I will start with a more math background to say, okay, you have a matrix like this, each row corresponds to a gene, each column corresponds to a cell. So because this is more closer to their background, but gradually, so then because you need to read a lot of papers in the field, as well as I think the fastest ways for them to learn is they will work together with clinicians and collaborators. So once they work with them together, so they will know more about the biological insights and what is needed for their research and what are the parts that their collaborators, they can leverage from their collaborators. So I think that's really helpful. And later, I think for a good biostatistician, I think is they will get a sense about what problems. So you can abstract from your collaboration into an interesting statistical problems. So I think throughout the years, so they will gain experience and learn from the collaborators. So I think how to identify a key problem. So it's a key thing. So that's the most important thing they need to learn from collaborators. Yeah.
Yeah, thank you. Because it's also remind the difficulty that we face in designing our curriculum, our own curriculum. To encourage. It's both ways, right? To encourage more statistics students to learn a bit more biology, or on the other way around, more biology students to have more numerical and computational skills. But thanks that you've done a great job. Okay, thank you.
Yeah, so we always welcome students from the whole Faculty of Science to take statistical courses from our department. So we extremely welcome all the students to come to our department. And also this year, so maybe some of you know that our department will actually have a new joint program with computer science that is called computational data science. So the idea is that we will also provide the training. For example, we have a stream called computational physics and also a stream called computational medicine. So we want students to receive the trainings from School of Life Sciences and Faculty of Medicine. So because knowing the background knowledge and knowing the subject is really important for them to dive in to understand the subjects and doing research. So I also hope like in the future, you can help with our students to do the final year project and also provide supervisions and also allow them to take your courses. So yeah, thank you very much.
Thank you.
Okay, thank KB for the question. So due to time limitation, I would like to skip my question and pass the time to the next speaker. So finally, please join me to thank Professor Wei again. Thank you.
Thank you.
Thank you, Professor Song and Professor Wei. So may I now invite Professor Li Wenjiang, Associate Dean for Research at the Science Faculty, to introduce our next speaker, Professor Chen. Professor Jiang, please.
All right, so it's a pleasure for me to introduce the next speaker, Professor Chen Manning. So Manning is an associate professor and director in the Earth System Science program, okay? And his research area is ambient air pollution, focused on the source and formation mechanism of particulate matters, okay? So he received his PhD in environmental science and engineering at California Institute of Technology. And he also did a postdoc in the chemical science division at Lawrence Berkeley National Laboratory. So please welcome Manning.
Thank you, Li Wen, for the nice introduction. Let me share the presentation. So thanks again for the nice introduction and also the invitation from the Faculty of Science. So in today's talk, so I'm going to talk about our collaborative research on organosulfates in ambient aerosols. There's some our team are interested in this topic because our team members think that organosulfates in ambient aerosol are a very important component. So they're very abundant in the ambient aerosol. And also they can have a huge implication on our environment and also our health. So in this talk, I would like to give just some quick introduction about what is aerosols. Maybe give you some example about their climate and health impact. And also again, so we want to address why our teams are interested in organosulfate in ambient aerosols. And also our team has tried to identify some unresolved issue or challenges in the atmospheric organosulfate studies. And also I would just want to share with you how our team members try to work together and then to address this complex issue. And then I will go to introduce some of our team members, some of the research themes that we identified, and also some of our initial progress. Okay, so what are aerosols? So of course, so aerosols, sometimes most of the time we heard about is the PM 2.5. So basically we are talking about the same substances. So aerosols, we are talking about some very small, tiny particles suspended in the air. So basically, usually we cannot see by our eyes. So you can see this is a figure show. So for example, for a PM 2.5, so what that means is that means the particle or the aerosol we say has a diameter less than 2.5 micron in diameters. And then you can also, this graph has a nice comparison. So you can compare to a human hair, and then you can see how small this tiny particle. So now you have some idea about what's the number here means. And of course, you can talk about PM10, PM1, or PM.1. So when you look at the size of the ambient aerosol, so on the right hand, the figures here, so you can basically also can see there's a huge size range. So we can have start from a few nanometers all the way to 10 micron. And of course, depends on the source of the types of the aerosols. They are not always spherical in shape. And also they can have a different kind of morphologies or shapes. So we can see this under the electron microscope. So these tiny particles can also have a huge impact on our climate. So of course, we know the aerosols can come from different sources. So some can come from human activities, and also some come from the natural sources. But the thing is, once these tiny particles present in the air, so they can absorb or scatter the sunlight. So we call this aerosol radiation interaction. And also more important is we need this tiny particle for the water vapor to condense and grow, and then to form the cloud droplet. So basically means that you see the terms called, we need the aerosol to sieve clouds, okay? And then also the clouds and also can have a huge impact on the radiation. For example, once you form the cloud, they can just reflect the sunlight back to the space. So this is what we call the aerosol cloud interaction. And then if you look at these graphs here, it shows the radioactive forcing of different species in the atmosphere. So of course, we know for the greenhouse gas, so we have a strong warming effect. So you can see here. And if you look at the aerosol effect, so look at the, say, for example, the aerosol radiation, the aerosol radiation interaction or the aerosol cloud interaction. So basically we have some ideas that we have what we call the net cooling effect. So therefore, nowadays, so people are very interested to study how the aerosol will affect the radioactive forcing, given we have a concern about the global warming. And of course, we know the ambient aerosols are bad for our health. Of course, in the past 2-3 years, so we're concerned about the COVID, the coronavirus. So I think we have the ideas, right, about the coronavirus. So this graph just shows you the size of the particle. And then if you look at the coronavirus, it's about 0.1 to 0.5 micron in size. So therefore, you can see this is, you can also consider this is a very small tiny particle can suspend in the air. And also at the same time, so they can press into some large droplet. So therefore, nowadays, it is known that the coronavirus can be, say, the droplet or airborne transmissions, okay? So now we have some idea about why we're interested to study the ambient aerosol. So we go back to our study. So why we are interested in organosulfates. So organosulfates, so basically we are talking about some organic compound containing a sulfate ester group. So here just some of the organosulfates have been detected in the ambient aerosols. And you can see from this graph is this kind of organosulfates is a kind of, we know it's called ambiguous constitute of the ambient aerosol. So basically you can detect this class of compound in all major regions or environments or different environments here. So this graph just show you the global overview of the OS concentration here. And also if you look at the mass or the concentration, so you can see in some regions, this class of compound, that means the organosulfate can contribute up to 50% of the organic mass, and also maybe sometimes up to 30% of the total aerosol sulfur content. So given their atmospheric abundance, so we expect that this kind of organosulfate can have a huge implication for the health and for our health and also the environment or climate. Okay, and but I also want to highlight here, so later we're going to show you that the number we show here maybe just still an underestimate or some lower limit. So there's some reason behind. So because we think that the organosulfate can have a huge implication for our health or environment, so there's been a lot of studies have been carried out to try to understand, say, the source, the formation, or the properties of this class of compound. So if you look at the organosulfate in ambient aerosol, so here I just want to show you that there's a number of organosulfate have been detected in ambient aerosol. Okay, so one way we can see detect this kind of compound in ambient aerosol is we can see from these figures is we can collect the aerosol onto some substrate or some filters. Okay, so we can collect the ambient aerosol on this substrate, and then we can use different types of technique. So one technique we can use is called this nanodesi. So basically, this kind of organosulfate can be easily ionized using the electrospray ionization technique in the negative mode, and then we can detect the ions in the mass spec. And then, of course, if we have a nice mass spec, and then we can assign the chemical formulas for the ions that we detect. And then you can see in the ambient aerosol, so we can detect a number of sulfur-containing ions. For example, you can have a number of CHONS or CHOS compound. Of course, not all of them are organosulfate, but we can also run additional analysis, like we can run some MSMS, and then we can look at the fragmentation ions. So that can also give us some more idea about the class or the nature of this kind of sulfur-containing ions. But in general, so you can see that in the ambient aerosol, so we detect a number of sulfur-containing ions or compound, okay, using this technique. And also, we can say that among these sulfur-containing ions, so nowadays we have some good ideas about hundreds of organosulfate can be present in the ambient aerosol. And then people try to understand their formation, their source, and also their concentration. But there's one problem is that, so even we have some idea about these organosulfate, but most of them cannot be positively identified. There's a number of reasons why we cannot say positively identify or quantify these kind of ions, this kind of species. So one reason I want to highlight to point out here is because we're missing some standard for detection and quantification. Here, I just want to show you one study. People try to identify or study the organosulfate in ambient aerosols. So this is the graph. They call this SOS precursor map for ambient aerosol. You can see that they can detect a number of organosulfates in ambient aerosol, and also they can classify them into different groups, depends on their origins or their sources. But the thing is, if you look at the figures on here, is to quantify these kind of ions or species. So we need kind of some of the standard, but you can see the table here just show that these are just some standard OS available. Oh, sorry. These are just only a few standards available to quantify or to try to identify, positively identify this kind of organosulfate. So therefore, the limitation, one of the major issues right now we have is we lack of this kind of standard for detection and quantification. And also if you go back to this map, and so you can clearly see that there's a number of organosulfates, it's called unassigned. So we have detected this, so we know this is some kind of organosulfate, but we don't know their chemical identities. And also because we have detected so many organosulfates in the ambient aerosol, so there has been a lot of studies try to understand the formation mechanism of this kind of organosulfate. So here, just a nicer summary. So nowadays, people have been proposed very different ways to form this kind of organosulfate. But the major idea is that we need some sulfate radical or sulfate combined with some organic compound to form this kind of organosulfate here. Here's just to give you two examples. So one is that you can have this kind of sulfur dioxide and then add into some double bond, and then you can form this organosulfate. And also you can see from these figures is you can have the isoprene, you do the oxidation, and then you can form some kind of a epoxide, and then this epoxide can be dissolved into the aerosol phase, and then have some reaction, or we call that ring opening, and then in the presence of the sulfate, and then you can form some organosulfate. So these are some of the mechanisms people have been proposed. But again, so there's a problem right now is that even we have been proposed many different formation mechanisms, but for the known mechanism, so far we know, so still we cannot fully explain the OS that we detect in the ambient aerosol. So therefore, nowadays, we still think that we are still missing some formation mechanism or precursor. But also our team find that most of the time right now in the literature or in our community, so people only focus on the formation of this kind of organosulfate. Here, just a graph show again. So you can have this kind of epoxide, you have the ring opening, and then you form this kind of organosulfate. But the thing is, the ambient aerosol can stay in the air for days, okay? So this is not surprising that once this organosulfate formed in the aerosol, they can further react, okay? But nowadays, our community, for some reason, they just assume, so once you form, this compound is very stable. But our team, our research team have a hypothesis and think that this is not true. So because if you're given, we know that the ambient is a very oxidizing environment, and also we have a lot of oxidants and also sunlight around. So we think that the organosulfate can further react and transform, and then you can form a number of new products. For example, here's some example for very important atmospheric process. For example, you can undergo some hydrolysis, maybe you can undergo photolysis or some kind of oxidation. So we think that this kind of organosulfate can further transform. And more important is that if you look at this graph, so nowadays people just maybe think about some first generation product or early oxidation product. But in the atmosphere, so we call chemical aging. So this kind of organosulfate can further transform and then form other product or some other new organosulfate. So we think that some of these we call unidentified organosulfate can actually come from the transformation of some early product here. So that's why this is, we also want to focus on this. And also for the modeling part, so people try to predict or understand the abundance of organosulfate in the atmosphere nowadays. But we find that most of the literature so far, or most of the models so far, is they only consider the organosulfate from only isoprene or just a few precursors. But of course, we know in addition to isoprene, so the organosulfate can come and can form from different precursors, like I show here. So it can come from isoprene, monoterpenes, or some VOC hydrocarbons. So, but all this mechanism has not been considered in current chemical transport model for some reason. So, and also I think the one of the reasons behind is because the mechanism is still not clear, and also some of the mechanism is not known. So therefore people just try to incorporate some well-known mechanism. So therefore the idea is that current chemical transport model is still insufficient. We think that it's still insufficient to accurately predict the abundance of the OS in the ambient aerosol. And also one more thing is, like I mentioned, because organosulfate is very abundant, so we think that it can also have a health implication. But nowadays, so we still don't know about the health implication of organosulfate, especially by inhalation. So of course we know that, for example, some of the species right in the ambient aerosol are toxic. For example, this kind of PAH toxicants. But we also have hypothesized that because we know some of these aromatic compounds can convert to become some of what we call aromatic organosulfates, and then they can present in the ambient aerosols. And then And then once we are thinking about is what happens if we have kind of have an inhalation of this kind of aromatic organosulfates. So what happens if you have some bioaerosol transformation? Okay, and then so what will happen? Okay, so we need to be affected our health or even more toxic than the parent pH and also the biological effects. So we're going to, this is certain unknown. So therefore, given these, so we try to form a teams to address these kind of topics. So basically, we can see from our team is we try to use the experts in field studies, organics, synthetic organic chemistry, chemical biological toxicology, MD or simulation, and also myself doing the laboratory studies here. And also our teams have identified some of the areas that we want to address. And also for this, at this stage, we want to focus on like some aromatic organosulfate because we can have a huge impact for our health. And also this can be formed in the urban polluted environment here. So here I just want to show with you that the different schemes, so what have been achieved so far. So for example, we have the scheme one will be led by Professor Yang from chemistry. So he was going to synthesize some new organosulfate standards. So we will target on some aromatic or nitroxy organosulfate. And we have achieved some significant progress. So we have been synthesized some new organosulfate standard already. And then we will target going to synthesize some of these new aromatic or nitroxy organosulfate standard here. And also with the standard, we're going to develop some new analytical methods so that we can better quantify the organosulfate here. So this is also want to show why we need to work together is because we have the synthesize that, and then you can have the standards so that we can use this for the biological study and also the laboratory study, and also with this standard and also the new development methods, so we can better quantify the organosulfates in the field study. And also the field study can tell us so what kind of organosulfate have been present, likely present in the ambient aerosol, and what kind of organosulfate we want to synthesize. So therefore, you can see this across the talk across the different theme. And also, we are going to conduct some field campaigns. So like I mentioned, we are focused on the aromatic organosulfate in the urban environments in Hong Kong and also the Pearl River Delta region. We have conducted some field campaign and also we're going to lead to the literatures to study the impacts. One of the ideas of course, we know people now know that the sulfur dioxide can play a significant role, but we have a hypothesis that like the different types of pollutant and also the oxidant can also play a significant role in the formation of the organosulfate in the polluted environment. So we're going to explore this in the coming field studies. And also the field study data can, like I mentioned, so can be important for constraining some of the model simulation and also together with the laboratory data, we can better identify the key factor governing the formation of these organosulfates here in Hong Kong and also the Pearl River Delta region. And also, like I mentioned, so basically now we don't know about the toxicity of organosulfate by inhalation. So therefore, we try to use some mice as a model to investigate the potential toxicity of organosulfate by inhalation. So this is some of the techniques that we have used. So we have set up this kind of chambers. So we have some mice, or you can also expose this organosulfate to some cells to study the potential toxicity. So one thing is that we want to test so whether the organosulfate, especially the aromatic organosulfate, are toxic. And also we also compare the toxicity to some well-known atmospheric compound that are toxic, for example, the pH. So one thing we want to test is whether this kind of the oxide, you can say this is kind of the reaction product, right, from this kind of the pH. So whether they will be more toxic or less toxic than the parent pH. So this is what we're going to study. So this study will be led by Jerome Hui from School of Life Science and also Professor Hokim Fai from Public Health. And then also myself, we have a laboratory step. So we're going to study the formation pathway. We'll continue to study the formation pathway of organosulfate. But we will also, like I mentioned, we'll more focus on the transformation. So we want to look at, so when this kind of organosulfate is further oxidized, so what will happen? Okay, so what will form the product? So, and also this will help to identify some of these organosulfates in the ambient aerosol. And then also we collaborate with Professor Steve Zhe from chemistry department to run some anti-simulation to really to look at some molecular details and also mechanistic understanding. So we have some ideas now is this kind of organosulfate is surface active, so they can likely accumulate at the surface. So therefore, it can play an important role in many, we call the multi-phase heterogeneous reaction or atmospheric process. And more important is that based on the MD simulation, we also want to look at so how this kind of sulfate as the group will affect like the properties, the kinetic, the mechanism, or the reactivities. And then finally, we just, because of the time, I rushed a little bit. But last thing I want to talk about is the air quality model. So this will be work together with the Professor Amos Tai. So we're going to run some state-of-art chemical transport model with some new formation and transformation scheme, and to say developed from our laboratory or MD simulation or the field studies. And then we try to improve the scheme and presentation so that we can more accurately predict the ambient abundance or environmental impact of ambient aerosol. So I just want to just have a conclusion nice. So here, so I'm not going to too details about the result, but just want to show you that this is a very complex issue, and then we also need to work together within the team so that we can have a better understanding about how these organosulfates will impact our health and also environment. And then finally, I just want to have acknowledgement from the support from the Faculty of Science. So thank you.
All right. Thank you very much, Manning, for a very nice talk. Now we open the floor for questions. You can just unmute and then ask that question.
Hi, Manning. Thanks for the nice talk. I have a general question. Since the pandemic, and then which has been 2 years, and then did we have a significant change in the in the emission or in the measurement of like the particles, like a PM 2.5?
Okay, thank you. So I think we also actually look at some data in the past 2 years. Because if you know the lockdown, if you look at the regional lockdown or maybe the lockdown in the whole country, so it only happens just a few months. So that's why we don't see a significant change, okay, or impact on the pollutant concentration. But one thing that's not related to the aerosol, but one thing is actually we find that is during the pandemic, actually most of the pollutant, the concentration of course, may drop due to the lockdown. But we find that the ozone actually can increase during the pandemic. This is because of some of the atmospheric chemistry mechanism. Because you reduce the NOx, but actually if you reduce the NOx emission, you actually can enhance the ozone formation. Yeah.
I see. Thanks.
I have a question, Manning. This is certainly very interesting and also very important research. When you get to the sulfate, you are actually, well, the sulfate will not be hanging in the air. They are more likely absorbed on the solid particles, where the solid particles is either the carbon-based soot or some type of a silica or inorganic compound based. So does that make any differences, or do you actually analyze that the base of where the organic sulfate are extracted from?
Yes, let me show you. So of course, we think that the organosulfate can form with different organic compound in addition to the soot or some black carbon.
Yeah.
You see the structure. Some of these structure here, maybe I show you the. So you can see the organosulfate is, of course, you can form, say, in the soot or some black carbon, but also you can see that just you can have some carbonyl or some unsaturated hydrocarbon present in the ambient aerosol, and then can say, for example, in this case, you can have a sulfur dioxide just add to this CC double bond. And also you can have this kind of epoxy, and then you have the ring opener, and then you add the sulfate group to this kind of organic compounds. So therefore, a number of organic compounds can potentially further oxidize and then form organosulfate.
They are oxidized before they are attached to the solid mass, or they are oxidized in the atmosphere and then become attached to the solid mass?
So for in this case, so for example, this ring opening epoxide mechanism, so the organosulfates in the aerosol phase. But you can see here is that sometimes the precursor is the sulfur dioxide can just react on the surface on the aerosol. But you are right, so most of the time is occur in the aerosol phase, not in the gas phase.
Okay. Thank you.
Thank you.
All right, any other questions? When you have a very quick question, you have 16, right? If you suggest, say, you in your next major grant application, you suggest to reduce to 3 stream and then try to integrate some of them together, what would that be?
Oh, okay. I think if we really want to narrow down the theme, so I would suggest at least that we need to keep the biological studies, because I think that would be more implication on clinical application. And then also, I think we also want to keep the field studies, and also we can combine the field study with the synthetics of organosulfate. And then, of course, I think it's also important to develop some new chemical transport model, I would suggest. If you really want to narrow down, so I think we need to keep is the field study modeling and also the biological studies, is to really want to narrow down the scope. Yeah.
Right. Thank you. So any further questions? If not, please join me to thank Yingying again for a very nice talk.
Thank you.
Thank you, Professor Chen and Professor Jiang. May we now invite Professor Chen to stay here. He is also the director of the Earth System Science Program, to introduce our next speaker, Professor Yang Wenfeng. Professor Chen, please. Okay,
thank you. So first, I'm very happy to introduce my colleagues, Professor Hongfeng Yang. Hongfeng actually joined CUHK in 2014, and now is an associate professor in our program, Earth System Science. And then Hongfeng is actively working on the earthquake source physics, subduction zone dynamics, fault zone structure, and also the evolution, and also some kind of we call the induced earthquakes. So therefore, you can see based on his research, so he has been committee members in several scientific committees in the Chinese Geophysics Society and also the Chinese Seismological Societies. And also he has been served in many editorial boards, and also now is also an associate editor for the Seismological Research Letters. And also in 2018, so that 2018, right? Then Professor Yang, also Hongfeng, received the Fu Changyi Young Scientist Award. This is a recognition of his great contribution to integrating the earthquake rupture dynamics and also the observational seismologies to advance our understanding of the earthquakes physics and also the hazard evaluations. And also I want to mention that Hongfeng is the first recipient of this prestigious award from Hong Kong. And also today he's going to talk about his research about the risk of the earthquakes induced by shale gas development in Sichuan Basin in China. So I think I'll pass the time to Hongfeng.
Okay, thank you, Yingying, for the very nice introduction. Now I start to share my screen. Can you see my screen now? All right, well, first I want to thank the faculty for the invitation to provide such a great opportunity for us to introduce some of our recent work and the progress. So as shown in the title, then I'm going to talk about some earthquakes, but then these earthquakes are related with developing shale gas. So I will explain later what is a shale gas. So, you know, everyone, you know, living here or for any other part of the world cannot live without the energy. So that is our reality. Actually, that is one of the grand challenges we are facing now as human beings. So as is shown in this chart, so our major energy sources are still from either coal, oil, or gas. And then in the, including some traditional biomass and then very small amount of some new type of energies. And then as we can see, as the time goes, and then definitely our need of the energy has been, vastly increased. So, so here, the major three parts, gas, oil, and coal, are all from underground. Therefore, earth science is critical. It's very important. So to ensure our energy supply. Unfortunately, they are mostly the fossil fuel. Therefore, although they provide the majority of the energy supply, but then they also dominate in the CO2 emission, and then playing critical roles in our climate change issues and global environmental issues. And as we can see in different parts of the world, and then the emission has been also greatly increased over the years. Therefore, it has been a critical issue that we need to sustain. We need to have our sustainable environment, but then also with this enough energy supply. Therefore, we are really in the dire need of the so-called clean energy. So what do we mean clean energy, which should be sufficient enough to produce a large amount of supply, but then in the meantime to reduce the emission as much as possible. So some typical examples are listed here include so-called hydropower. For example, we can build a dam and then to utilize the gravitational energy change and then to use the water to generate the power for us. And then we can also utilize a geothermal, which is right now a rapidly increasing new type of energy source. The idea is very traditional because we know inside the Earth, the temperature is increasing over depth very quickly. So if you can go down by 1 kilometer, then the temperature may increase 20 or 30 degrees. So if you can pump some cold water in and then suck the water out, and then they will become hot. Therefore, we can utilize the so-called geothermal energy. And then it turns out in Guangdong and also even in the Greater Bay Area that there are a large amount of reserve for such kind of a geothermal energy, which can be developed in the future. Another part is I listed here is so-called natural gas. Actually, in I guess most families, and then we are using, and then in the kitchen, that is actually one of the energies. So their common attributes include, of course, providing critical energy supplies, but another important attribute, common point, which is important for us, and also the focus of my talk today, is that they can all induce earthquakes. Some of them can induce very large ones. For example, like in Guangdong, Heyuan, which is only 200 kilometers away from Hong Kong, in 1962, and there was the largest actually reservoir-induced earthquake in Xinfengjiang, which is just 1 kilometer away from this dam, 2 years after the construction of this dam. And then recently in South Korea, there's an earthquake, a Pohang earthquake with magnitude 5.5, and that is due to the geothermal project. And also recently in Sichuan Basin, we found a series of magnitude 5 larger earthquakes, which is associated with the shale gas development. So what is shale gas? So in general, we need the reservoir. We need to extract the energy from underground, and the energies are extracted from so-called traditional reservoir. In that sense, the rock formation has a larger porosity, and so that also a lot of cracks make it more permeable. Therefore, you can just utilize the flow, natural flow. There, we can, suck out all the oil and gas in the formation. But then after those, kind of a depletion in most conventional reservoirs, and then has been actually long recognized, there are a lot of resources in such type of rocks in this black color. It's called shale. But then as just from our eyes, we can see these type of rocks are more, condensed, and then with a very small porosity, and so that's a low permeability. Even though in the past that people know there are a lot of energy reserves in these type of rocks, but then it is really difficult, extremely challenging to get those. So in the past 20 years, engineering techniques have been developed greatly. With one of the critical development is that we used to be able to drill down vertically, but then in the past 20 years, and then they start to drill down horizontally. So after you drill down a few kilometers beneath the surface, and then you can drill along some formations over there. And then over here, then we can just inject this additional fluid to break such type of tight rocks to create the fractures so that the energy, the gas can be sucked out. So this technique is so-called fracking. So later, then I may use this term to represent this process. And then has been well known, this technique has been really effective, has been used worldwide. And then it turns out, once you found, oh, this is an effective way to get the energy, and then it becomes inevitable because of the large amount of market. But then on the other hand, this process may induce earthquakes, as I mentioned before. And also such injected fluid may migrate underground and causing potentially significant environmental problems. So these are something, we have to think about and then start to investigate. So talking about the earthquakes, and then I want to just briefly mention some mechanism which will be important for us to understand some later part in the slides. So earthquakes occur on faults, and then we know the earthquakes actually it's just a product of the fast fault slip or the fault movement. All right, during earthquakes, our ground can move at the rate of 1 meter per second, just close to the speed that we can run as a human being. And we know there are different mechanisms. One direct way, if you inject the fluid, for example, to conduct the fracking to extract the natural gas, if there is a fault nearby, the fluid may go into the fault, therefore lubricating the fault plane, causing the fault to slip fast to generate earthquake. This is a common mechanism as we recognize. But the reality is more complex because first, a lot of small scale faults are invisible, or we don't know there are faults over there. So before really an earthquake occur, making it really difficult to evaluate the seismic hazard. Another thing is even fluid may not directly go into the fault. There are other mechanisms which may possibly induce earthquake as well. So making it even more challenging to think about if we want to mitigate the potential risk for such human induced earthquakes. So here I want to move into Sichuan Basin and then taking the examples over there to demonstrate how we are doing and then how we try to possibly tackle some of those questions. So in Sichuan Basin, there are a rich reserve as shown in this map, as shown in those blue colors. And there are a lot of shale gas reserve found over there. And by far, there are some developing shale gas fields with this one called the Weiyuan and Rongxian counties, and which is the national demonstration base with the first vertical well. And then this is the typical platform where you may see the fracking being conducted. You see a lot of trucks over there with large water tanks, and then they're pumping the water down to the ground. So here, let's look at the shale gas production in China over the years. So China has not developed shale gas until 2009, as I showed in the past picture. Production started around 2013 also. And then as we can see, the production now in billions of cubic meters has been really, increasing a lot. But then if we look at this figure at the lower panel, the earthquake numbers in the Sichuan Basin has been also drastically increased. And showing in this red curve, which showing not only the numbers, but also the magnitude of the earthquakes being larger and larger, being more significant. So these earthquakes by far are mostly concentrated in three parts. This part is Weiyuan and Rongxian. And this part is called the Changning shale gas field. And this part is a newer one, just having a magnitude 6 earthquake last year, called the Luzhou shale gas field. So I will be talking about this part as my focus. All right, that part become my focus because in 2019 February, there were 3 earthquakes occurring in consecutively 2 days. One of the earthquake occurring on February 25th and killed 2 people and then caused 12 injury, which become actually the first earthquake in the shale gas field in the world that caused fatalities. That also caused frustration among the local residents. And then we conducted some research to combine the geodetic technique and also seismological investigation. We identified the earthquake to be a very shallow one, and then the results has been reported by local media and also international media among different channels, and becoming one of the significant events in the Sichuan Basin shale gas field. And then later we conduct additional effort, but unfortunately, the COVID started by late 2019, so it's significantly put down the barriers for us to go to the field, including myself. But then I really, want to thank my team in the Shenzhen Research Institute, which make it possible to deploy additional observational systems in the field. And then these are some field photos that we take as our team members who went to the field in 2020 and 2021, and then to monitor additional earthquakes over there. And with additionally collected data, then we apply the state-of-the-art machine learning technique in identifying the earthquakes so that we can delineate with high resolution the spatial and temporal evolution of the earthquakes. As shown in this cartoon, so you can see each black dot representing one earthquake. And then as the time goes, and they just pop up as different places. And here is one red line. This is a well-known fault in the field. So the critical feature is that these earthquakes are not concentrated along this fault, which is normally expected if they are natural, because earthquake could not usually just occur on fault. So if we just look at the earthquake distribution with details, we can see a lot of small scales lineations over here. And then they are actually turns out to be coinciding with some fracking wells with the orientations in the region. Later we identified in the field. So there's no doubt most of the earthquakes are induced by fracking activities. But the question is, how can we possibly offer some useful advice to ensure the production and safety operation in the production? We also compare with most of the earthquakes with the, we call the stratigraphy, different colors meaning the different type of rocks and age underground down a few kilometers. We can see most of the earthquakes are concentrated between 3 to 6 kilometers also. Again, with those large black lines indicating the local faults over here, you can see most of the earthquakes are not occurring on these faults. All right, so they are associated with human activities. So we also conducted our detailed study on the largest earthquake occurring in the region with a moment magnitude 5 that occurred in September 2019 to integrate our seismological and geodetic technique. So here the advantage using the geodetic technique is that we can show the satellite image over here and with very clear location of the earthquake. And then we also know how the fault is oriented underground and what is the slip distribution over there. And then with all the support from the seed money from CRIM's project, we can conduct the previous work, and then with a focus on the seismological investigations on the earthquakes over there. But really, some other objectives in our CRIM project also include investigations on the role of the science in earthquake policy and governance system. because the science for inducing earthquakes might be common among different regions, but how we may possibly implement the policy for the local residents and to live with those induced earthquakes might be different. All right, so this is really one of the objectives for us to continue. Unfortunately, this has to be done with some field surveys to collect, how the local residents will react, but then we cannot go to the field because of the travel restrictions. But then also we tried one with RIF last year. That was shortlisted, but unfortunately that was not eventually funded. But then we got some support from NSFC, which is actually a smaller scale CRF over there with our support from the Shenzhen RI Research Institute in Shenzhen. So here I just want to highlight induced earthquake is actually not a regional problem, but a global issue. And then it actually demands collaboration among the different units, including government and industry and also academia. And also this is not only just limited to shale gas, but also to any other type of clean energy like hydropower, geothermal, as I mentioned before. And even just to pinpoint to the mechanism inducing earthquakes, which is very challenging and complex. Here I want to utilize one example. And here are two platforms where the fracking was conducted, and which are just 4 kilometers away from each other. If we look at this platform, it's 37, and when it is conducted over here, and then during the operation of this platform, we can see a lot of earthquake occurring over here. So definitely induced directly by the fracking. But then if we look at the operation period for this platform, which is here, but then you can see the earthquake numbers are quite few. And then there were no direct earthquakes associated with this fracking. So only 4 kilometers away. In earth science, it's just like, like a molecule scale for us to think about. But then really very different behavior. But then shortly after this operation of the fracking on this platform, the largest earthquake with magnitude 5 occurred. So really challenged us to think about how the different mechanism will apply, and can we possibly provide some precursory information for the safety in operation. All right, so that requires a more than seismological investigation, and at least we need to conduct a reliable geomechanical modeling, which has been done in the previous project with Ting Fang and Liu Lin, my colleagues, and myself, with successful applications. And then here I want to move it to such shale gas investigation as well. So we try to, we also submitted one CRF this year, and then we hopefully get the proposal fly, and then we got the different components with seismological investigation, geodetic, and rock physics. And then we also look for colleagues who are potentially interested in such, earthquake problems with statistical input and with some mathematical model, media with fluid flow, so that we can construct the reliable geomechanical models and to advance our understanding over there. So with that, and then I want to conclude with this slide, is that, again, we do need energy, and we are also needing clean energy. But the question is how to get the clean energy or the gas over there, and then by mitigating the earthquakes in the meantime. So there are a lot of earthquakes induced by such kind of activities. Can we do something better to ensure our energy supply, but then reducing the risk of earthquakes? All right, I want to stop here and thank you again for the great opportunity to introduce our work. Thanks.
Thank you, Hongfeng, for the very nice talk. So may I ask if there are any questions for Hongfeng from the audience?
May I have a question?
Yes, please.
So Hongfeng, this is very important and also not only on the geophysics, but closely related to the global energy development. As I came from the Pennsylvania in the US. Just before I left the Penn State, Pennsylvania was the largest shale gas production base in the United States, and to the point where it was called the fourth industrial revolution in the US, because it changed the US from a energy importing to energy exporting country. And because of shale gas, the light oil production was also substantially increased, and by the same technique. I think the earthquake certainly is one of the big concern in many states, yes, even between the Pennsylvania and the neighboring state, the New York state and New Jersey state, where they take a different stand about whether they should proceed on shale gas. And the major concern is earthquake or the seismic movement. And I, as a sort of a chemist, I dealt with the solid intercalation compound, where you have multiple layers of a compound, where one of the layers in the middle can actually become a lubricant that help you to exfoliate the compound to two separate layers. So I wonder whether in different sedimentation layers, when the water can actually swell into the middle layer somewhere, it could cause a sliding between the different sedimentation due to the sort of the gravitational forces. Could that be a cause of the seismic or the earthquake as well?
Yeah, thank you, Professor Song. That is really a great question. So as shown in this plot, and actually it has been proposed that, you know, among the different layers, among the different colors, and if we inject enough fluid, which may possibly cause the sliding of this layer against this layer, and which may also cause earthquakes over there. But then if there's no additional, you know, injection of the water, these layers should be just contacted with each other and then very stable. So that is one of the scenarios over there. But the challenge for us is that these, you know, layers are very natural nearly everywhere on the earth, and with mostly are stable. And then how we may possibly evaluate the potential risk of whether or not this part of the layer may become, you know, unstable. So that is really one of the challenging points.
Yeah.
Yes. That's a great question.
Thank you. And actually, just a follow-up, one of my friends at Penn State, Derek Aerosols, he has, well, at around the time before I left Penn State, he has a study, or started a study, where they would do the dry gas injection versus a water injection.
Yeah.
If you do that, and then the gases that you inject would not cause a swelling of the sedimentation, and therefore, one thing that is save a huge amount of water, because water, the amount of water use, energy water nexus issue, over 75% of water use in global water use is actually in the energy production utilization in this energy cycle. So by not using water, you can save water and improve the water issue. But there is other one. If you use a dry gas injection, could you prevent earthquake?
Yeah, I think this is a great idea, which is still in the stage that is under, I would say, the pilot stage. So it has been proposed to use the dry gas to replace the water, to inject over here to create a fracture. And also it has been proposed to use some CO2, supercritical state CO2, to replace water, to reduce the water usage, but also to store the CO2 over there. All right, so, but these are still under mostly the laboratory stage, as far as I know. And in the industry operation, still water has been still the majority source for conducting such kind of activities. But this just opened up a related question, which is also very important, because to respond to the potential climate change, and CO2 sequestration has been proposed to basically to store the CO2 underground. And it has been also suggested to utilize the fractures created after the fracking and then producing the natural gas, and to inject the CO2 to store them over there. But CO2 sequestration has been done in different parts, and which turned out is also be able, it's also able to induce earthquakes. So that becomes another issue for CO2 sequestration. Yeah, but this is a great question and related to the induced earthquake society as well.
Thank you. Thank you.
So maybe any other question for Hongfeng? Okay, if not, then maybe let's thank Hongfeng again, and then I will pass the time back to the science faculty office.
Thank you.
Thank you, Professor Yang and Professor Chen. So may we now invite Professor Jiang to share the closing remarks. Professor Jiang, please.
All right. So it has been really good half day. I think thanks for all these excellent speakers. I've learned a lot. Okay. I would like to say thank you to Angela, also Jackie. So to conclude, may I invite Din Song to provide to deliver concluding remarks? Din Song, please.
Well, I thought when you're going to give the concluding remarks.
Oh, in the programs, you. Yeah.
Okay. First of all, I thoroughly enjoyed every presentation today, and I learned a lot. I hope that all of the participants have learned something new and beyond their normal daily learning and research within their own disciplines. And I think this is important to get to know something outside your field. It's just like having the antenna in your mind, absorb something from other side, and then build into the knowledge framework. And that often is the start. Oh, I wonder whether we can do this, whether we can team up that. And this is actually critically important. And I think over the years, I led about 80-some projects and nearly over 20-some collaborative projects and multi-million-dollar projects, largely because of this type of a learning and conversation with people from outside my own field. That's very important and very helpful. And so I hope that we all learned something. And the Faculty Research Day really is organized to support our people, support the advance of the research through this type of cross-disciplinary discussions. In that sense, I hope we all benefited, and this will provide some motivation for further collaborative work. As we mentioned earlier, our claims will continue, and our next round of solicitation is already out, and we just selected several projects for the current round, and then the solicitation continue to be out for other collaborative, and people from different disciplines should get together and talk about some potential interests, and not only from this Faculty Research Day. This serves as an example to catalyze the discussions and promotion for interdisciplinary collaborations. So with that, I want to thank Li Wen and our faculty research panel and our science faculty staff for spending a lot of time and effort to planning this event. And I want to thank Professor Qin to give this inspiring talk, and all of our speakers today and your research team that you represent for the presentation today, for your collaborative work, and that has resulted in significant advances, as we can all tell from the individual presentations. I'm very happy to see the advance in the collaborative work, and I look forward to see even further development in the faculty collaborative work, leading beyond CUHK and even beyond Hong Kong and reach out globally. And thank you, and I wish you all a great day for the rest of the day, and hope this will catalyze the germination of the salt for collaborations. Thank you.
Thank you.
Thank you.
Bye
Bye.
Speaker.
Thank you.
Yeah. And then, yeah. Bye bye. Thank you.
Bye.
Okay.
Yeah,
Okay.
thank you.
Bye.
Bye.
Bye.